Hello, welcome back to Bioclass Bites. In this video, we are going to talk about meiosis and its stages. Don't forget to subscribe to my channel and like and share this video. So for the first part of this video, we are going to talk about gametogenesis, specifically oogenesis and spermatogenesis. In the second half, we will actually go to the main topic, meiosis and its stages. So the word meiosis was actually taken from the Greek word meion, which means to diminish, okay? So this refers to the reduction or the diminishing or the reduction of the chromosome number from the parent cell to the daughter cells, okay? So we define meiosis as the cellular process that results in the number of chromosomes in sex cells. Again, sex cells. Meiosis is exclusively performed by gametes or sex cells in the body. So in humans, meiosis is performed by the egg cells found in the female ovaries and sperm cells found in the male testes. Okay, so all the cells in your body, they perform mitosis as discussed in the previous video. Brain cells, skin cells, lung cells, pancreatic cells, kidney cells, they all perform mitosis. So all the cells in your body, all the body cells perform mitosis. Only your sex cells, so egg cells for females and sperm cells for males, only those kind of cells perform meiosis. So meiosis is the cellular process of cell division in sex cells that involves a reduction division or reduction process by which each pair of homologous chromosomes pass on to each daughter cell. So there's a reduction of the chromosome number into which the daughter cells will just perform regular mitosis afterwards. Okay, so this is an overview of meiosis. As you can see, there are two stages of cell division. Unlike in mitosis, there's just this, okay, from one parent cell into two daughter cells. But in meiosis, there are two stages of cell division. The first cell division is actually the reduction division, in which the chromosome numbers are reduced into half, okay? While the following cell division are just regular mitosis. However, instead of producing just two daughter cells, the products are four daughter cells. And instead of producing diploid daughter cells or those with equal number of cells, the products are four haploid cells. So you can see here diploid parent cell. So when we say diploid, 2N, it has two copies of the chromosome number. 2N. N here represents the chromosome set, okay, of the parent cell. So it has two copies. But right after cell division, the first cell division, the first reduction division, there's now two haploid. So usually you have here 1N, but in other books or resource, just letter N. It means that they only have one copy of the chromosome set. Okay? So two haploid cells, and then these haploid cells will just undergo regular cell division, wherein the haploid chromosomes will further divide into haploid daughter cells, as you can see here. So yeah, so the product of meiosis are 1, 2, 3, 4, 4 haploid daughter cells. So in animals, meiosis always results into the formation of gametes. So that's the other term for sex cells, gametes. While in other organisms such as plants and some fungi, it gives rise to spores or spore formation. Now, what makes meiosis important as early as now? I want to emphasize this. Meiosis is important for sexual reproduction. Okay? Because here, there's a union of egg cell and sperm cell. So one gamete comes from the father, another gamete comes from the mother. It is important and it occurs in all eukaryotes, including unicellular organisms that reproduce sexually. Because here you have the differentiation of the genetic donors, male and female. So this is meiosis is an important process of sexual reproduction. So, again, not to emphasize the difference between mitosis and meiosis, So somatic cells, those are your body cells. Somatic, soma, body cells. Um, the cells, the parent, the resulting daughter cells are always diploid. When we say diploid, D, di, two, they have two copies of the chromosomes, or two of each type of chromosomes, two copies. So body cells, somatic cells, they divide by mitosis. So in humans, the resulting daughter cells are also diploid, 2n, or that's a total of 46 chromosomes, okay? While in meiosis, these are performed by gametes. Those are your sex cells, sperm, and egg. So the resulting daughter cells are haploid, half. They just have one copy of each type of chromosomes. And they divide gametes, sex cells, they divide by meiosis. So in humans, the product daughter cells are haploid, so they have 23 chromosomes. So I recommend that you visit this Cells Alive animal cell meiosis animation. So it will discuss all the different stages of meiosis. So I'll provide the link in the description below. This one also a short video, but it will summarize everything we will talk about meiosis. Again, I'll provide the link in the description below. And this one, meiosis, plants and animals, a longer description, but a longer discussion, but still more important, okay? Also very important for you to watch. Meiosis in plants and animals, I'll provide the link in the description below. Since meiosis is the cell division of gametes, I think it's also important that we talk about gametogenesis, okay? So which is the process by which diploid or haploid precursor cells undergo cell division and differentiation to form mature haploid gametes, okay? So basically this tells us how egg cells are produced in females and how sperm cells are produced in males. General overview, as you can see, similar to a typical meiotic division. There are two stages of cell division here. So the precursor cell or the major parent cell in oogenesis are called primary oocytes. And then after the first cell division, they are now called secondary oocyte. However, only one out of four daughter cells will actually mature into an egg cell, while the rest will degenerate as polar bodies. Okay, so they will not be used. They will not mature into an actual egg cell. So as you can see, one primary oocyte gives rise to one mature egg cell. That's quite different in spermatogenesis or in sperm cell production. So the precursor cell is the primary spermatocytes, and then after the first cell division, they are called secondary spermatocytes, and then after the second cell division, they are now called spermatids. So this will now mature into your sperm cells. But as you can see, for every one spermatocyte, you produce four spermatids, which will become four sperm cells. Okay, that's why in terms of numbers, we have more sperm cells than egg cells. So again, so this is a more detailed discussion of spermatogenesis and oogenesis. We will go through them one by one. But basically, you start off with a diploid spermatogonium, which will undergo meiosis. So this one are found, so in spermatogenesis, the spermatogonium are found in the testicles of the baby boy while he was still inside his mother's womb. So they will undergo rapid cell division, rapid mitosis. But however, with the onset of puberty, the rest of the spermatogenesis process will continue. So spermatogonium will now become a primary spermatocyte, still diploid. The secondary spermatocytes, so there are now two new daughter cells, and then those secondary spermatocytes will become haploid spermatids, and then following cell differentiation, all those four spermatids will become future spermatozoa or sperm cells. While in oogenesis, so these are the oogonium, so these are precursor cells that are found in the ovaries of the female when she was still inside her mother's womb, which will undergo mitotic division. Then those primary or diploid primary oocytes will undergo the first cell division, and then that cell division will give rise to one secondary oocyte and the first polar body. Okay, so this one will no longer grow into an ovum or an egg cell. So this one, the secondary oocyte will again divide, second cell division, second meiotic division, which will give rise to one ootid, haploid ootid, then your second polar body. While the first polar body will further divide into two new polar bodies, then that ootid will actually differentiate and mature into a haploid egg cell, while the second, third, and fourth polar bodies will just degenerate. So they will not be utilized anymore. So for every one oogonium, you have one ovum. For every one spermatogonium, you have 4 sperm cells. So we've talked about this already in male reproductive system, okay? So again, we define spermatogenesis as the process by which male spermatogonia develop into a mature spermatozoa or sperm cell. So this is the diagram of a mature sperm cell. We've talked about its parts. Its main cell body, then it has this midpiece that contains a lot of mitochondria, and then the tail that allows it to propel in the female reproductive system. So spermatozoa is the mature male gametes, and in many sexually reproducing organisms, and spermatogenesis is the male version of gametogenesis. So we've talked about this, that spermatogenesis actually is triggered in puberty and can continue uninterrupted until the death of the male organism. So however, there will be a slight decrease in the quantity of produced sperm cell as the male ages. Okay, so very important because spermatogenesis is highly dependent upon highly conditions for the process to occur correctly, and it is important for sexual reproduction. So together with spermatogenesis on the onset of puberty, there's also the development of the secondary male sex characteristics. We've talked about this: facial hair, broad shoulders, increased body hair, maturation of the genital organs in males, and having a more muscular body. So to explain further of the different steps of spermatogenesis, I recommend that you watch this video, Human Physiology, Reproduction, Spermatogenesis. I'll provide the link in the description below. Also, this video, Spermatogenesis, again, I'll provide the link in the description below. So again, just to give emphasis, so spermatogenesis is actually triggered during the onset of puberty because during this time, the testes secrete large amount of testosterone. So testosterone, the male hormone, a steroid hormone, actually stimulates the growth of the testes. So the growth of the testes here, and that initiates maturation of the seminiferous tubules. So spermatozoa is then released into the lumen of the seminiferous tubule to be transported into the epididymis, where they are stored. Now, during ejaculation of the sperm cell, they are propelled through the vas deferens and they exit out of the urethra, together with the secretions of other glands that form the fluid semen. Okay. So the triggering factor here is puberty for spermatogenesis to continue. So I recommend that you visit Encyclopedia Britannica to have more information about spermatogenesis. Again, I'll provide the link in the description below. So what's the importance of spermatogenesis? Why is it important that this must actually occur without mistake, okay, properly? It is because spermatogenesis produces the sperm cell, okay? Produces the male sperm cell, which will then fertilize the egg cell or the oocyte, which will eventually create a new individual called, which will start from the zygote. So it's important that half of the genetic material of the zygote must be chromosomally normal, okay? So this is the cornerstone of sexual reproduction, since there are two gametes contributing to the new organism. half of that, under spermatogenesis, the sperms must have chromosomally normal um, sperm cells to produce chromosomally normal zygotes. Um, because any mistakes in spermatogenesis can actually create chromosomally abnormal zygote. Okay, examples of those. So for example, in humans, we have a total of 46 chromosomes. So if something went wrong in spermatogenesis, that could create, instead of having a 24 chromosome sperm cell, 23 chromosome sperm cell, it could produce a 24 chromosome sperm cell. And when it fertilizes an egg cell, it could have, instead of 46, a 47 chromosome zygote. And sadly, that could actually create a lot of problems for the baby, for the offspring. So examples of those would be um, Down syndrome. So instead of having 46 chromosomes, they have 47 total of chromosomes. Other genetic disorders such as Turner syndrome, Klinefelter syndrome, they have unequal or incorrect number of chromosomes in the zygote. So starting with spermatogenesis, this process must produce chromosomally normal sperm cells that will fertilize the egg cell in the future. So oogenesis is the female version of spermatogenesis. So same with males, it's also during puberty that the process of sperm oogenesis are further happens. Okay, so together with the hormones, luteinizing hormone and follicle stimulating hormone, they stimulate the production of sex hormones by the ovaries. The ovaries then will produce estrogen and progesterone, which will begin the bodily changes that could happen during puberty. So that involves maturation of that involves the development of secondary female sex characteristics such as maturation of breast, broadening of hips, development of pubic hair, and of course the menstrual cycle. Okay, so to understand better how egg cells are developed through oogenesis, I recommend that you watch this video entitled Human Physiology, Development of the Ova. I'll provide the link in the description below. And this video, oogenesis, how the female reproductive system produces eggs, again, I'll provide the link in the description below. So this is how oogenesis happens. Okay, we've talked about this a while ago. So, when the female was still inside her mother's womb, she has a lot of oogonia, which undergo regular mitotic division. However, at the onset of her own puberty, the rest of the meiotic cycle, meiotic cell division will happen. Okay, so here we've talked about this from one primary oocyte. After all the meiotic division, it will produce one egg cell. Okay, so yeah. So we've also talked about the female reproductive system. So oogenesis happens in the ovaries. Okay, so, and then should fertilization happen, it must happen along the fallopian tube. And then when successful, the zygote will now embed itself in the uterine wall, and then it will grow into a new fetus. We've also talked about the hypothalamic pituitary ovarian axis back in female reproductive system. So it shows us what's happening in the ovaries, what's happening with the hormones of the body, and what happens to the blood in the uterine wall, okay, in the uterine lining, in the progression of a 28-day menstrual cycle. Okay, so we've talked about this thoroughly in the female reproductive system. If you still have, if you have forgotten, you can visit that and review this part. Okay. I also recommend that you visit Encyclopedia Britannica for more information about oogenesis. I'll provide the link in the description below. And how about importance of oogenesis? Okay, so if sperm cells must be chromosomally normal, same thing with egg cells. Okay, so egg cells, oogenesis is the production of egg cell. And another thing that's important here is that oogenesis contains all the materials needed to initiate and maintain metabolism and development of an offspring. So only the sperm cell only contributes the nucleus. Okay, it only contributes the genetic material. In addition to forming a haploid nucleus, oogenesis also builds up the cytoplasmic enzyme, the mRNA, and the organelles and metabolic substrate. Okay? So while the sperm cell becomes differentiated mostly for motility in order to reach the egg cell in the fallopian tube, the egg develops a remarkably complex cytoplasm to contain all of this for the future zygote. Okay? Another thing to note, um, this part actually um, um, should fertilization occur, only then will the ootid eventually develop into a mature egg cell or mature fertilized egg cell. But if there's no fertilization, most of those egg cells are actually arrested, or they are not continued in terms of cell division. They're arrested at their at metaphase stage, so they do not continue with anaphase 2 and telophase 2. Um, and those will be further discussed in the videos recommended. Okay, so what happens to those arrested egg cells? Okay, so together, what's the importance of egg cells, of oogenesis and spermatogenesis? So I keep on mentioning this. It's important that those two processes happen successfully because each gamete must have only half of the usual number of chromosomes. They must produce haploid sperm cells because they will anticipate fertilization in the future. Okay? They will anticipate that those haploid sperm cells will fertilize a haploid egg cell. Okay? So if that is not what will happen, it could result into serious abnormalities in the offspring. Okay? I've mentioned this a while ago. Chromosomal abnormalities can arise from incorrect spermatogenesis, such as Down syndrome, Klinefelters, and even spontaneous abortion. So if you have more than 48, 49 chromosomes instead of having 46, the fetus or the embryo will be naturally and spontaneously aborted. Nature will not allow such abnormalities in the chromosome number to continue. Okay? The offspring will not survive. Okay? Most chromosomally abnormal zygotes will not survive long after conception. So it will be naturally aborted. If they happen to survive conception, they could have disorders such as Down syndrome and Klinefelter's syndrome. So this diagram shows us the stages of the human life cycle, and it also shows us the interaction of mitosis and meiosis in order to perpetuate life. So we also similar to our discussion in sexual reproduction, if you still remember that video, we all start with a haploid sperm cell and haploid egg cell. Okay, those are products of meiosis. Okay, so meiosis produces gametes, eggs, or sperm for reproduction. So the male gamete is haploid; it only has one set of chromosome. Same with female. Upon fertilization, upon union of these gametes, they will now unite to form a diploid zygote. So it's now 2N. It has two sets of chromosomes. Okay? So that zygote, so N one set, 2N two sets. So that zygote will undergo mitosis, rapid mitosis, many successful mitotic division inside the mother's womb until it becomes an embryo, and then the embryo becomes a fetus. Then when the baby is born, it grows, and throughout its life, it will continue to undergo mitosis. Okay, so the mitosis produces cells for growth and repair of that growing baby, of that growing individual, until he reaches puberty. Okay, until he reaches puberty, wherein his testicles will then trigger the continuation of spermatogenesis, which will then produce the haploid sperm cell. So if she happens to be a female, then during on the onset of puberty, then her ovaries will now continue oogenesis to the from oogonia to maturation of the egg cell, okay, until it produces this haploid egg cell. So in the future again, when they unite, they form another zygote, which will now undergo mitosis until it grows up, and then during puberty, his his testicles or her ovaries will continue undergoing meiosis to produce haploid gametes. Okay, so this is the human life cycle through meiosis and mitosis. To further differentiate mitosis and meiosis, I recommend the very entertaining video from Amita Sisters, Mitosis and Meiosis Side-by-Side Comparison. I'll provide the link in the description below. So at this stage, we'll now look closely into the different stages of meiosis. Okay? And there are some difference from mitosis. So I've mentioned that in meiosis, there are two meiotic division or two cell division. So during meiosis 1, so the first meiotic division is called meiosis 1 or meiotic division 1. The second division is meiotic division 2. During meiosis 1, what's important about this is that it separates the homologous chromosomes to produce two haploid cells. So separation of homologous chromosomes to produce haploid daughter cells. So this is why it's called a reduction division from diploid to haploid. So a regular diploid human cell contains 46 chromosomes, so that's 2n. However, after meiosis 1, it now becomes 1n, or they only have 23 chromosomes. Okay, so they are considered diploid, or they have a total of 46 chromosomes because it has 23 pairs. So 23 times 2, 46. So 23 pairs of homologous chromosomes. During meiosis 2, so this is actually a cell division similar to mitosis, whereby the sister chromatids are finally split to create 4 haploid daughter cells. So this is just a normal mitotic division performed by 2 cells. Okay? However, the difference here between mitosis, aside from the total number of daughter cells, is that these cells will no longer return to interphase. Okay? They stop here. If these are sperm cells, they mature into spermatozoa, and then they are discharged out of the male body. So they no longer return to interphase, into gap1, S phase, gap2. If this is female, oogenesis, they stop here. Oogenesis stops here. The three polar bodies are no longer are discarded. Then the female ovum, if it is fertilized, will then become an egg cell. If not, this will that unfertilized egg cell will be discarded out of the body together with the blood coming from the uterine lining. So the products of meiosis no longer return to interphase. They are discarded out of the body. So these are the phases of meiosis 1 or stages of meiosis 1. So they still follow PMAT. They still follow prophase, metaphase, anaphase, and telophase. However, just to differentiate, since this is meiosis 1, we add Roman numeral 1 here. Prophase 1, metaphase 1, anaphase 1, and telophase 1. So what are the events during first meiotic division? Okay, so during prophase 1, so here, the important thing that happens here is that the pairing up of homologous chromosomes. So again, we have 46 chromosomes. It's actually made up of 23 pairs. So those 23 pairs will look for each other and then they will pair up. This does not happen in mitosis. Okay, they just line up along the middle, all 46 of them. But here in prophase 1, the homologous chromosomes are paired, okay, while the rest are still the same. So disappearance of the nuclear membrane, appearance of the spindle fibers and centrioles, they happen. That's what happens there, okay. So after prophase 1 will be metaphase 1, wherein the homologous pair of chromosomes will line at the middle, okay. So instead of just one chromosome, so you now have 2 chromosomes here. And then during anaphase 1, the homologous chromosomes will then be pulled into the opposite direction. The sister chromatids stay together. Okay? And then during telophase 1, everything returns to normal. So that's the appearance of the nuclear membrane, the disappearance of the spindle fibers, and then disappearance of the what do you call this, of the centrioles. Okay, now one thing that you have to remember that makes meiosis very important is that during prophase 1, during the time that the homologous chromosomes pair up with each other, that is where this event crossing over happens. That is when the homologous chromosome exchange genetic material. Okay? Which is why, as you can see here, you start off with this purple and then red, just call it red chromosome. But after crossing over or exchange of genetic material, you now have a red chromosome with a violet part here. And here you have a violet chromosome with a bit of red chromosome here. So they exchange genetic material. So they exchange genetic combination. And this is the reason why we've talked about this in sexual reproduction. The products of sexual reproduction are unique individuals, unique organism, and this is the cause for that. This is why even if you and your siblings came from the same parents, you are not clones of each other, because at meiosis, during prophase 1, during pairing up homologous chromosome, there has been this exchange or reshuffling of the genetic profile, genetic material of your parents. So this happens both in the sperm cells and in the egg cells. So right after meiosis 1 is meiosis 2. So same, no? They also undergo PMAT. However, since this is meiosis 2, we put here the Roman numeral 2. Prophase 2, metaphase 2, anaphase 2, and telophase 2. So if you will notice, these are very similar to mitosis. Okay, so the starting cells are now considered haploid. Okay, so during prophase 2, the nuclear membrane also disappears, the chromosomes condense, spindle fiber appears, the centrioles appear. During metaphase 2, two cells, okay, you must show two cells. Two cells, the chromosomes align at the middle, okay, at the metaphase plate. And then anaphase 2, the chromatids, sister chromatids are pulled apart. They separate towards the opposite ends. And then telophase 2, the newly formed gametes are now haploid. They just have one copy. So there's also the reappearance of the nuclear membrane, disappearance of the spindle fibers, and then eventually it will lead to cytokinesis. So it will produce 1, 2, 3, 4 haploid. They just have one copy. 4 haploid egg cells or sperm cells. Okay? So, what's the importance of meiosis? Again, I keep on mentioning this. Meiosis ensures stable sexual reproduction. Okay? The reason why oogenesis must produce 23 chromosome egg cells and spermatogenesis must produce haploid 23 chromosome sperm cells is that in the future, should fertilization happen, then 23 plus 23, they will produce 46 chromosomes in a zygote. So that's a chromosomally normal offspring. Without halving of the chromosome count, fertilization will result in zygote that have twice the number of chromosomes. So for example, if there's no reduction, division, so the product of meiosis are 46 chromosomes in a sperm cell and 46 chromosomes in an egg cell. So when they unite, that gives you how many? 92 chromosome in a zygote. That's not possible. Nature will not allow that to happen. It will be spontaneously aborted. Okay? It will not survive. So they must produce another 46 chromosomes. Okay, we've talked about this. They must produce 46 chromosome zygote. So 23 here plus 23 here to create 46 zygote, diploid zygote organism. Right? Okay. So another thing, because of recombination, because of the crossing over that happens here, okay, crossing over, the exchange of genetic material, recombination and independent assortment of homologous chromosomes allow for greater diversity of genotypes or genes in a population. So this is again the reason why we are not clones of each other. Products of sexual reproduction are not clones, but they are genetically unique from each other. Okay? And this allows us better survival. Just like what's happening right now with COVID, no? Some people have survived because they might have a good genetic composition that results into a good physical composition that allows them to fight off the virus, while some possibly because of not so viable genetic combination, it did not allow them to survive the virus. Okay, so having greater diversity in a population allows the survival of the species. So to learn more about meiosis, I recommend you visit this lesson from Khan Academy about meiosis. I'll provide the link in the description below. And to clarify more about the chromosome number during meiosis and mitosis, I recommend this still from Amoeba Sisters chromosome numbers during division, demystified. I'll provide the link in the description below. So basically, this is what that video will just tell you. Okay, so how many chromosomes will be produced after meiosis and mitosis? So in mitosis, okay, you start off with a diploid parent cell. Okay, however, after DNA replication during S phase, you still have 46 chromosomes, but you will have 92 chromatids. So chromatid, that's just one structure like that. But when it's a chromosome, that's the X structure like that. So it will undergo mitotic division, prophase, metaphase. However, during anaphase, there will be the split, okay? Splitting of the chromosomes into opposite sister chromatids. So there's a total now of 92 chromatids in the nucleus until cytokinesis. So telophase, the nucleus is still dividing, but after cytokinesis, once there's a reappearance of the nuclear envelope in the two opposite daughter cells, now it returns to a diploid number into 46. Compare that with meiosis, okay, wherein you start off with 46 chromosomes, okay, but because of the pairing of the homologous chromosomes, so there's now 4 chromatids, okay, so there's a duplication of chromatids, okay, but right after the first meiotic division, it will now result into daughter cells having 23 chromosomes or 46 chromatids. Okay. Now this is not yet finished. Okay. The cells will now undergo second meiotic division, wherein the number will be further reduced into half. So from 23 chromosomes or 46 chromatids, after prophase 1 and metaphase 1, they will now be separated from each other, but the cell is continuously elongating. Then it will continue until cytokinesis. Now, in the 4 daughter cells, they will now just result into having 23 chromosomes. Okay, this is why in mitosis, the products are 46 diploid daughter cells, while in meiosis, it produces 23 chromosomes in haploid daughter cells. So this is just a simple pop quiz. So let's try to define the following terms. Very easy. So cell cycle, we define that as the series of events that take place in a cell leading to its division and duplication. So in prokaryote, that's just binary fission, while for eukaryote, it involves interphase and cell division. So again, interphase, that's the phase wherein the cell grows during gap 1, accumulate nutrients, duplicates its DNA during S phase, and then prepares everything for cell division during gap 2. So cell division is the process by which one parent cell divides into 2 or more daughter cells. Mitosis is the formation of 2 nuclei. Again, mitosis is the division of the nucleus performed by body cells to produce two diploid daughter cells. Meiosis are performed by sex cells, wherein the parent chromosome will produce four haploid daughter cells. So these are more different comparisons of mitosis and meiosis. So in terms of chromosome set of the mother or parent cell, you both start with diploid, okay, in mitosis and meiosis. What type of cells perform mitosis? Body cells, such as lung, muscle, brain, internal organ cells, while meiosis are performed by sex cells, egg and sperm. Total number of daughter cells after mitosis, 2 daughter cells. After meiosis, 4 daughter cells. Chromosome set of the daughter cells in mitosis, diploid. Okay? They are just the same as their parent cell, while in meiosis, haploid. Okay? They just have half of the chromosome set. In terms of purpose, mitosis is used by cells for growth, repair, and asexual reproduction in some prokaryotes or in some organisms. While for meiosis, that includes the reduction of the chromosome number from diploid to haploid for sexual reproduction. So meiosis is for sexual reproduction. So this one is actually, just easy. You're supposed to draw the following stages of cell division, but the answers are here now. So for anaphase, this is how it looks like. Anaphase 1. So as long as there's number here, it refers to meiosis. So anaphase 1, you see here the separation of the entire chromosome. So in metaphase 1, they are found here at the center, the homologous pair. But anaphase 1, they separate from each other. You see here the results of crossing over. Prophase, so there's no numeral, no number here, so it means that it is a mitosis stage, mitotic stage. So it involves the condensation of the chromosomes, disappearance of the nuclear envelope, appearance of the spindle fibers, and centrioles. Metaphase 2, so you have here the idea, it's a meiotic division. So metaphase 2, so here's the alignment of those homologous, of those chromosomes at the middle, and then they will divide that way. Okay? For your reflective journal log, I mentioned that it's for mitosis, it's important that cells undergo proper cell division. If they do not, that could lead to cancer formation. So please watch this video on cancer from healthy cell to a cancer cell. I'll provide the link in the description below. That ends our video. I hope you learned something new today. Don't forget to subscribe to my channel and like and share this video. Till next time, goodbye.